Now the four kingdom classification system. Four kingdom classification system, it was given by the scientist Copeland. Copeland created a new kingdom. The name of the kingdom is Mycota. But later on the name Mycota was changed to Monera. So the kingdom which was generated by Copeland, that is Mycota, Mycota name was later changed to Monera. But the name Monera, term Monera was not given by Copeland. It was given by the two scientists, that is Doherty and Allen. Now the kingdom Monera, it is booked for only prokaryotic organism. Yes. Just now we have discussed in the prokaryotic organism, well-defined nucleus is absent. So kingdom Monera, it is booked for only one group of organism, that is prokaryotic one, and in this prokaryotic group there comes the bacteria. तो इसका मतलब सबसे पहले बैक्टीरिया को डिफरेंशिएट करने वाला, सबसे पहले बैक्टीरिया को अलग करने वाला, इट इज नॉट हैकल, इट इज कोपलैंड. After the four kingdom classification system, now we are going to deal with five kingdom one. Five kingdom classification system, it was given by the scientist R.H. Whittaker. The new kingdom which was created by Whittaker, that is fungi. Already four kingdoms were there: Plantae, Animalia, Protista, Monera. And now that new kingdom which was created by Whittaker, that is fungi. So new kingdom which was created by him, that is fungi. Now we have to see what were the different criteria which were utilized in the five kingdom classification system. The different features which were utilized for this classification system. Five kingdom classification system is mainly based upon five criteria. First criteria is complexity of cell structure. Complexity of cell structure. That means whether the organism is prokaryotic or it is eukaryotic. Yes. Whether the organism is prokaryotic or it is eukaryotic. Second feature which was utilized, that is complexity of body organization. Complexity of body organization, which means whether the organism is unicellular or it is multicellular. Unicellular or multicellular. Now the third one, that is mode of nutrition, which is main criteria of his classification. Mode of nutrition, that is whether the organism is autotrophic or organism is heterotrophic. If the organism is heterotrophic, then the three categories they can be there. Three categories. Whether the organism is parasite, showing parasitic mode of nutrition. Parasitic mode of nutrition means organism is deriving food from other living organism. Got it? Organism is deriving food from other living organism, and you have studied in the previous classes this living organism is known as host. Second is saprophytic. Now, in this case, organism is deriving nutrition, organism is deriving food from dead and decaying organic matter. Next one is holozoic nutrition. Holozoic nutrition, it involves engulfing of solid food. Yes, it is ingestive nutrition, ingestive mode of nutrition, that is engulfing of solid food. Next feature, that is reproduction. Reproduction, whether the organism is showing asexual mode or it is showing sexual mode of reproduction or showing both the types of condition. The last criteria which was not utilized in any of the classification system till now. Yes, that is phylogenetic analysis or evolutionary relationship of organism. Now, what do you mean by the evolutionary relationship? Evolutionary relationship is that how the new organisms, they are derived from pre-existing one. Yes, how the evolution has occurred. How the new organisms, they are derived from pre-existing ones. Which organism is primitive and which organism is advanced. So here, primitive and advanced groups. So here, primitive and advanced groups, they were distinguished. Yes, so these are the 5 criteria on the basis of which Whittaker gave 5 kingdom classification system, and amongst these 5 criteria, which is the main criteria for classification? This one, main criteria for classification, right? So now what I am going to do, I am going to perform comparative study of 5 kingdom with respect to certain characteristics. So now comparative account of 5 kingdoms with respect to certain characteristics. Monera, Protista, Fungi, Plantae, Animalia. So we have to perform comparative study between these 5 kingdoms on the basis of certain characteristics. First characteristic which I will deal here, that is cell type. Monera, just now we have discussed they are going to have prokaryotic cell condition, prokaryotic cellular organization. Prokaryotic, for prokaryotic I am only writing PRO, and in rest of the kingdoms there is presence of eukaryotic cellular organization. For eukaryotic, please use this short form. Now after the cell type, now we will see what is the condition with respect to cell wall. Second feature, cell wall. In the case of Monera, cell wall is present. Cell wall is present, but mind it, this cell wall is not cellulosic in nature. Not cellulosic. The cell wall is made up of other polysaccharides. Along with that, there is presence of amino acid, which we will discuss in detail in the forthcoming lectures. Protista, some of the groups, cell wall is present. Fungi, cell wall is present, but it is non-cellulosic. It is non-cellulosic. You might be knowing cell wall is made up of chitin. Planting, now cell wall is present. All of us we know it is cellulosic in nature. Animalia, cell wall absent. Now after this, now the next feature, that is presence or absence of nuclear membrane. Monera. Monera comes under the category of prokaryotic organisms. And in the prokaryotic organisms, well-defined nucleus is absent. Nuclear membrane around the genetic material. Genetic material, all of you know, that is DNA. Around the genetic material, nuclear membrane is absent. Prokaryotic group, nuclear membrane absent. And in all the eukaryotic group, nuclear membrane is present. Yes. After the nuclear membrane, we will see body organization. Monera, generally the organisms which are present in this group, they are unicellular. Write UNI for uni. Protista, strictly unicellular and eukaryotic organisms. Fungi, mainly multicellular. Yes, mainly multicellular. But the true tissue formation is absent. There is formation of loose tissue. Yes, do you know what is tissue? Tissue represent group of cells performing common function. But here true tissue formation is absent. There is presence of loose tissue, which we will learn. This loose tissue is known as mycelium. Plantae, tissue level organization present. Along with that, there is presence of organ level organization. Animalia, tissue level organ level as well as organ system level organization. Yes. So do you know what is organ? Organ is representing group of tissue performing common function. Then what is organ system? That is aggregate of organ or group of organs performing common function. Now after this, now the last feature, that is mode of nutrition. Now the last one, nutrition. Monera, the organisms, they can be autotrophic, write A for autotrophic, as well as heterotrophic. In the autotrophic, there are two categories. Organisms which have the ability to synthesize their own food by utilizing light energy. They are known as photoautotrophs. Photoautotrophs. Organisms which can synthesize their own food, but not by using light energy, by using chemical energy. So they are known as chemoautotrophs. Both the conditions are there. In the heterotrophic, either the organism is parasitic, saprophytic mode of nutrition, holozoic mode of nutrition in the Monera is absent. Now coming to the Protista, autotrophic as well as heterotrophic. In the autotrophs, they are photoautotrophs. Right? In the heterotrophs, they can be parasitic, saprophytic, as well as showing holozoic type of nutrition. The most diverse type of nutrition is present in Protista. सबसे डाइवर्स न्यूट्रिशन जिस किंगडम के अंदर प्रेजेंट है, दैट किंगडम इज प्रोटिस्टा. नाउ कमिंग टू द फंजाई, हेटेरोट्रॉफिक, कैन बी parasitic as well as saprophytic. Plantae autotrophs. Which category? Photoautotrophs. Animalia, heterotrophs, can be parasitic, can be holozoic. Yes. So the different features, they are very much clear to you in different groups of the organism in the 5 different kingdoms. Okay. Now after the 5 kingdom classification system description, now we are going to see some of the shortcomings of the system. Now from this classification system, what are the positive points of the classification? That was very much clear. There was distinguishment between autotrophs, heterotrophs, unicellular, multicellular. But here now we are going to see what are the drawbacks which are associated with it. Whittaker classification system divided the algae into 3 different kingdoms. Yes, he divided the algae into 3 kingdoms. One is Monera. Under the Monera, the algae which was placed, that is BGA, known to all of you. What is the full form? Blue-green alga. It is a prokaryotic algae. That's why by utilizing the system of Whittaker, now it is placed under Monera. second kingdom where the algae was placed, that is Protista. Protista के अंदर place करी है unicellular eukaryotic alga. Yes, just now we have discussed Protista is booked for only which group of organism? That is unicellular eukaryotic. So the algae which was placed under Protista, that is dinoflagellates and diatoms. Now the last group where algae was placed, that is Plantae. Under the Plantae there was placement of eukaryotic algae which is generally multicellular and includes green alga, red alga and brown alga. Got this? So utilizing the Linnaeus classification system, all the three algae, they were placed together in Plantae, but Whittaker classification system divided the poor algae into three different kingdoms. Got this? Now the next drawback, viruses, they were not provided with any position. No position was given to the viruses. No status was given to the viruses. Now after 5 kingdom classification system, now let's start with 6 kingdom. Sixth kingdom classification system, it was given by the scientist Carl Woese. Carl Woese, what he did, he divided Monera, that is prokaryotic kingdom, into 2 kingdoms. Yes, what he did, he divided Monera into 2 kingdoms. That is primitive bacteria, archaebacteria, advanced bacteria, that is eubacteria. Yes. So he divided Monera into 2 kingdoms, that is archae and eubacteria. Now what was the characteristics utilized for this differentiation? What was the basis of differentiation? One of the bases of differentiation is cell wall. In the case of eubacteria, cell wall is made up of peptidoglycan. Throughout the lecture while teaching bacteria, I will call this peptidoglycan as PG. The peptidoglycan wall, it is also known as murin. But in the case of archaebacteria, the cell wall structure was different from eubacteria. Cell wall structure is different from eubacteria. Cell wall consists of the component, but these components, they are slightly different from the murin. That's why you say that in archaebacteria, cell wall is made up of pseudomurin. Second feature, that is cell membrane. Now you have studied this in the previous classes. Structure of cell membrane. It is made up of 2 main components, that is phospholipid and protein. Phospholipid and protein. Let's see in the eubacteria. Let's say this is a phospholipid. This part of the phospholipid, it is known as head. It is water loving hydrophilic. Right? This part of phospholipid, these 2 parts, they represent tails. tails, they are water repelling, that is hydrophobic. So this is one phospholipid. Membrane is made up of the phospholipids, and these phospholipids, they are arranged in the form of bilayer. They are arranged in the form of two layers like this. This is one layer and this is second layer. Bilayer arrangement. So you can see the bilayer arrangement. Always the heads are towards outer side and tails are towards inner side, that is interior. But what about the archaebacteria? In the archaebacteria, again these phospholipids are present. Again these phospholipids are present, but now these tails of two layers, they are joined together. Yes, the tails, they are showing linkage. The tails, they are showing linkage, due to which instead of the bilayer appearance, now it is going to give the appearance of one layer, that is monolayer. Yes, so now it is monolayer arrangement. Got this? Along with that, the fatty acid chains which are present here, yes, these are tails, and these tails, they are made up of fatty acid. Got it? Tails are made up of fatty acid. Fatty acid tails, in this case, they are unbranched. Fatty acids, they are unbranched, and in this case, they are branched. So, on the basis of these features, Carl Woese divided Monera into two kingdoms, that is Archaebacteria and Eubacteria. Got this? Now, the Carl Woese, he performed genetic analysis of different organisms. Yes, he performed genetic analysis of the organisms which are present in the different kingdom. He performed genetic analysis by utilizing the character, by utilizing the chemical, that is 16S ribosomal RNA gene. Feature which was utilized, that is 16S ribosomal RNA gene. Now what is gene? Gene is going to represent DNA which has coded information to perform specific function. Yes, it has coded information. It is our DNA which has coded information to perform specific function. This might be known to you that DNA, it has the information to synthesize RNA. DNA has the ability, it has the coded information to synthesize RNA, and this synthesis, it occurs through a process that is known as transcription. RNA is of three types. tRNA, that is transfer RNA. mRNA, that is messenger rRNA, which is ribosomal RNA. And what he has used? He has utilized such type of DNA which has the ability to form ribosomal RNA. And the ribosomal RNA which it will form, that is 16S, where S is going to represent sedimentation coefficient. Got this? Again, I am telling you here, gene is DNA. It will form RNA. The gene which was utilized here, that gene has the ability to synthesize ribosomal RNA of what size? That is 16S. So he has studied DNA sequence. He has studied gene sequence for the classification purpose. After studying the gene sequence, what he concluded, that these 6 kingdoms, they can be grouped under 3 categories. Yes, they can be grouped under 3 categories. I am going to write the name of these 3 groups. One is archaea. Second is bacteria. Third is eukarya. After the genetic analysis, he generated these 3 groups. Yes, these 3 groups he called them as domains. Till now, what I have told you, which is the highest category of classification, that is kingdom. But above the level of the kingdom, one more category which I am discussing here, that category is domain. Now, in the domain archaea, the kingdom which is present, that is archaebacteria. In the domain bacteria, it is kingdom which is present, that is eubacteria. The last one, that is eukarya. It is going to have all the kingdoms which are eukaryotic. That is Protista, fungi, plantae, and animalia. Yes. So it means Carl Woese gave, on the basis of genetic analysis, how many domains? 3 domains. And how many kingdoms? That is 6 kingdoms. Now, according to the Carl Woese, all these 3 groups, they have originated from same ancestor. ये तीनों ग्रुप से इनका जो ओरिजिन हुआ है वो सेम एंसेस्टर से हुआ है. तो इट मीन्स ऑल दीज 3 ग्रुप्स दे हैव कॉमन ancestor. And what is the name of this common ancestor? That is progenote. So just like the Whittaker classification system, this classification system is also based upon evolutionary relationship. So this is all about classification system. Here we have studied 2 kingdom, 3 kingdom, 4, 5, and this was the last one, 6 kingdom classification system. Now in the biological classification chapter, I will deal with Monera, Protista and fungi in detail. So first of all, let's start with Monera here.